and I'm delighted to be here tonight because I'm going to tell you about the human genome, and I think it's critical to understand what this means for every individual in this world. So we're going to talk about genetics, and I want to just impart a few principles of what the human genome has done, and in order to do that, I got to talk a little bit about genetics, but in a very sort of personal way. Now, one of the good fortunes of my lifetime is that I grew up in San Francisco in the 1960s. And while that was a great time for music, it was not a good time for cancer. I'm a pediatric oncologist, and I treat children with cancer. And at that time, in the 60s, a child with leukemia, the most common childhood tumor, had about a 10 to 15% chance of living 5 years from diagnosis. Thankfully, over the past 40 years, that has improved to over 85%. That dramatic improvement was done through the development of agents to eradicate cancer. It wasn't based on a knowledge of what was driving the cancer in the first place. Now, you got to go back in time because in the 50s, it was thought that viral agents were the root cause of cancer. And the reason for that was in the animal kingdom, most animal cancers, tumors, if you have cats, the feline leukemia virus is caused by viruses. But in the 60s and 70s, actually it was probably in the 60s, a dramatic turn took place when it was recognized that when tools to analyze chromosome structure were developed, that analyzes the chromosomes. Remember, you have 23 pairs of chromosomes in your cells. But when people looked at cancer cells, they saw aberrations in the chromosomes of the cancer that was not in the person. And this set off the whole field of cancer genetics. And that's what I'm going to tell you about tonight. Now, it's all about personalized medicine. That is understanding the reason for a patient to be cured or fail therapy is based on three variables. One, of course, is the tumor. One is the host itself. How does a patient take the medication? How is the drug metabolized? Does it get to the target? And finally, what does the therapy use? And this complex interaction here is what results in cure or treatment failure. Now, to start with the genome, you got to understand genetics. Now, it's really not that hard. Remember, the genetic code is embedded in the chemical structure of DNA. It's a double helix, and there are 4 letters that establish the code. That's like a bead on a chain. It's the DNA helix. Within the human genome, there are 3.2 billion base pairs. Remember, the Human Genome Project was started in the late 90s. It was thought to be over-budgeted. It was criticized. What was completed ahead of time, and the first sequence was developed in the year 2002, and the human genome was completed in 2006. And it was determined that the 3.2 billion base pairs here, but only 2% of the genome encodes genes that make messenger RNA that make protein. So the flow of genetic information is DNA, RNA, and protein. And of course, it's the protein that matters because that's the workhorses. That's the enzymes like insulin. It's the enzymes that regulate glucose to tell cells when to divide. It's the proteins. But the proteins are encoded by the RNA, which is encoded by the DNA. So remember this flow of information here. Now, as I told you, the human genome was a phenomenal achievement. It was done ahead of time and under budget, like no other NIH endeavor. It contains 3.2 billion base pairs. Now, what was really interesting is, remember, the first sequence of the human genome, 70% of that information came from one individual. But when people began to sequence the human genomes of different people, to their surprise, that we're 99.9% identical to one another. You pick any 2 people in this world, regardless of race or ethnicity, you're 99.2% identical to one another. Now, in that 3.2 billion base pairs, as I told you, only 2% really seems to matter. Now, when the human genome was being sequenced, the genomes of fruit flies and roundworms were sequenced first. These are very low organisms, and they were determined to contain 25,000 genes in fruit flies. So given the complexity of the humans, it was thought that humans must contain at least 6 times the amount of genes compared to fruit flies. Now, a very existential recognition occurred when it was determined that humans, like fruit flies, flowering plants, only have 25,000 genes. But the difference between a human and a plant is that humans can take the same genes and they can shuffle them to make about 10 times the number of proteins. So all the genes are the same, they can shuffle them around to make an assortment of proteins. I said 10 times, it's 3 times. Now, it turns out that if you're an evolutionary biologist, it is kind of interesting that I told you that each person in this room is 99.9% identical to one another. If you compare your genome to a mouse, you're about 20% different. If you compare yourself to an orangutan, you're only 1% different. There's only 1% difference in the human genome that distinguishes us from the orangutan. And one of the biggest benefits of the genome has been in terms of evolutionary biology, you can compare relatedness amongst different species now, and it's changed dramatically the whole order of molecular taxonomy. So that's some cocktail facts there for it. Now, I told you that we're at 99.9% identical to one another. Now, how many people have heard of SNPs? So pharmaceutical companies now and everybody are after these things called SNPs, because I told you that we're about 0.1% different from one another. And these minor differences do occur, and there are about 1.4 million of them located throughout the genome. They're just a single base difference. I told you the 4 bases. A single base difference is called a single nucleotide polymorphism. But these SNPs, despite their minor variations, can cause critical differences in the way a person responds to a medication. Because I'm telling you, remember, this is a gene right here, right? And the gene codes for RNA, and it makes a protein down here. And I told you that the code, if I change this code by one base here, it may change this protein by 1 amino acid. If that's insulin, it can change the function of your insulin by about 2 to 3%. So it can change the function. So you have the same protein with a subtle difference. It can account for variations in your enzymes. The best example, there are about 20 enzymes that degrade carcinogens in the environment. So we're exposed to carcinogens every day in what we eat, sunlight, and we have enzymes that break down those carcinogens and make them inactive. Now, my enzyme, because I have one SNP, may be about only 98% as good as yours. So I'm not going to be quite as good as breaking down those carcinogens as you may be. And that maybe put me at a slightly increased risk for cancer. Now, you're not likely to pick this up if you look at broad populations, because that subtle difference may be only a minor variation. But there's no doubt that some of the risk for cancer may be because of these SNPs right here. It's been shown that people, in terms of with asthma, that respond to asthma medications, they may not respond quite as well because these SNPs change some of the enzymes in their body that interact with the medicines that we give for asthma and diabetes. So drug companies, we want to look at this because you may be able to match the drug with the SNP to get a better outcome. And this is actually being done now because the same genes that degrade carcinogens also degrade certain drugs. And people who have a certain SNP may metabolize the drug a lot quicker, and therefore it may be less effective. So you can match the SNP with the drug and determine the dose. Any questions about that? I want to just make sure that comes across. That's a SNP, a single nucleotide polymorphism. It's that 0.1% difference between us that may account for why we respond to drugs a certain way or why we may be susceptible to carcinogens. But it's a hot topic. Okay, when I talk about cancer is a genetic disease, and when I tell you that, what I'm talking about is not an inherited disease necessarily, but when I compare the tumor genetics to the host. So remember, it's only one cell in your body that develops a mutation that converts that cell to a cancer cell. There are probably about 5 different steps. But when I compare the normal DNA in your body to the cancer cells have a corrupted DNA sequence. They've developed mutations that change that cell's behavior. So on one hand, I talk about the host, that's what I'm talking about, the SNPs, but we also want to understand the tumor. So both these factors relate to genomic medicine. So when cancer cells first became, when we developed the techniques to look at cancer cells and to look at their genetics, it was clear that cancer cells are corrupted. And their genetic information is corrupted in any one of 5 ways if you look at cancer cells. So if this is a gene right here, let's say this is a growth gene. This gene, in your white blood cells, it could be a gene called c-MYC. So when you get an infection and your white blood cells sense a pathogen in your blood, this gene gets turned on and it tells your white blood cells to begin to multiply, to neutralize that virus or bacteria. So that growth switch gets turned on. But then, of course, when the invader is neutralized, that growth switch has to get turned off and your white count goes back down again. Well, the same gene in cancer cells can be amplified. That is, at its location, it can be doubled and tripled and amplified 150-fold, so that it gives an increased signal all the time. So instead of having this switch, the cell has a growth signal all the time, regardless of whether the virus is in the environment or else. That's called amplification. That's seen in breast cancer tumors with HER2/neu. It's seen in a childhood tumor called neuroblastoma. It's a very common way that the cancer cells may corrupt normal growth signals. You may take the same gene and mutate it to give the protein a novel biological property. The best example is a gene called RAS. It's seen in 90% of pancreatic cancers. It's seen in a number of colon cancers and lung cancers, probably one of the most common genetic mutations in cancer. RAS is another growth signal. So this mutation turns the growth signal on, and it's incapable of turning itself off. Finally, you may take 2 different genes and different parts of the genome and splice them together. And that spliced together creates what's called a chimeric protein that also takes on novel biological properties. Now, the interesting thing about this translocation is unique for the cancer cell and not shared by normal cells. And I want you to remember that, because that may be the Achilles heel of cancer cells. And finally, there's another group of genes that are like the brakes. I'm going to describe it on the next slide. And these brakes tell the cells to slow down and stop dividing. So when the organ gets a certain size or begins to impact on a neighboring tissue, the brake goes on and tells the cells to stop growing. When cancers, these brakes are deleted. The cancer gets rid of the brakes to endow it with this growth accelerator. So in one case, we get the increased signal, the abnormal signal. This gives growth-promoting properties of the cancer cell, and no signal means the brakes are lost. And it brings up the concept, there are 2 different types of cancer genes, okay? Now remember, the big breakthrough occurred in the 70s with the first development of what's called recombinant DNA technology. And they found out that one of these genes, right here called an oncogene, was corrupted in human cancer. And that changed the whole field. I remember I was at Stanford reading an article on this, and I was convinced, I was a clinical doctor, that I needed to do genetics to cure childhood cancer. But these oncogenes, under normal circumstances, in normal cells, are growth promoting. They turn the green, make the light green. They tell a cell to grow, to multiply, and they're activated in cancer inappropriately. They get mutated so the switch is on and it doesn't get turned off. That's an oncogene. That's like the RAS oncogene. It's like HER2/neu. These are oncogenes. The other type of genes is what I told you before. These are tumor suppressor genes. These are the brakes. It tells cells to stop. They send suppressive signals to the cancer cell. They tell it to stop dividing. But in cancer, they're inactivated. The cancer cells kick them out. So it's a balance between oncogenes and tumor suppressor genes in the cancer, not your normal cells. Now, there are some inheritable forms of cancer, undoubtedly. But what I'm talking about now, it's in the cancer that's corrupted. Any questions about that? Oncogenes, tumor suppressor genes. And there's about 300 of these that have been identified now. And they fall into certain pathways. So this is what I'm talking about. In a normal cell, there may be a growth factor in the environment. It could be a signal like a virus, or it could be a growth factor. and it binds to a receptor on the cell surface, and it sends a signal to the cell to start dividing and multiplying. It could be during embryogenesis, or it could be during infection. But once the growth factor is not there, this thing turns itself off. When a cancer cell, what happens is these growth factor receptors get mutated. So that even in the absence of a growth factor, they send a signal to the nucleus and it can't stop growing. It's mutated. It's addicted to this growth signal. And likewise, in the same model where a growth factor binds to the receptor and sends a signal, cells are scripted to make sure there's a lot of quality control. So the single growth signal by itself is not enough to set the cell off under normal circumstances. There's a lock here that needs to be unlocked in order for this signal to manifest itself in its entirety. This is the tumor suppressor genes right here. So, but the tumor suppressor genes is not going to unlock that unless it sees a lot of signals. So what happens in the cancer cell is that this lock is lost. So now the same growth promoting property takes place because you've just deleted the brakes. That's what cancer is all about. It's a corrupted genetic information in the cancer cell. Now these mutations, under many circumstances, happen by accident. You can increase your chances of those mutations occurring, obviously, with sunlight, smoking exposure. You can increase your chances, but many times these mistakes happen by accident. Now, it turns out that you need 4 or 5 of these to occur to convert a normal cell into a cancer cell. That's why only 1 cell out of the trillions in your body becomes cancerous, because it takes many steps. You can increase your chances of that occurring. But in childhood, for example, we know it's by accident. It was not a case of environmental exposure or things like that. In adults, it's a different story. But it's 5 or 6 mistakes. And it turns out now that we know in all of us, every day these mistakes are occurring. And we have repair mechanisms to repair the DNA. Sometimes, even with the best of repair mechanisms, one of those mistakes slips through. And if it happens to hit the wrong switch, things become activated. So if you're over 50, like myself and others in this room perhaps, if you took my blood right now, I have a 30% chance of picking up a mutated oncogene in my blood, even though I'll never develop cancer. I'm sorry. I thought I turned that off. Let me get that back. So it turns out these initial steps in cancer stem cells are probably occurring all the time, but they're aborted in our body because it takes, like I say, 5 or 6 steps to become cancerous. Now, there's no doubt that the therapy we use today has been curative for a large number of people, cancer chemotherapy. But let's face it, in 15 years, we don't want to be using chemotherapy because its damage is relatively indiscriminate. So conventional chemotherapy works on dividing cells by basically damaging it. So all the drugs we give today, most of them are targeted against the DNA. And since the cancer cells are growing, they're very dependent, on the DNA. And all these cells, things like antimetabolites like 6-MP and methotrexate, Taxol, vincristine, adriamycin, cytoxin, they all work on damaging the cell. And it's called a therapeutic index. It hits the cancer cell most, but it causes collateral damage on normal cells. That's called a therapeutic index, the difference between effectiveness and toxicity. In antibiotics, it may be quite wide. You can give a large dose, and the toxicity is relatively low. But with cancer chemotherapy, it's a narrow therapeutic index. The difference between cure and causing a lot of damage is narrow, so you have to dose it very precisely. It's like throwing a grenade into a foxhole. A lot of collateral damage. This is not targeted therapy. It's effective. It's not specific for cancer cells. Cancer cells are uniquely susceptible, but normal cells are affected, and there are side effects. What we want is targeted therapy. Now, it turns out that these mutations generate targets in the cancer cell that aren't shared by normal cells. So therefore, they're tumor-specific. And they become a focus now of a lot of work to target those mutations for 2 reasons. One, it's these mutations that are driving the cancer. So for the first time, we've been able to look under the hood of the cancer cell, and we know what's driving the cancer cell. It's an amplified HER2/neu. It may be a mutation in the EGFR receptor for lung cancer, but we know these are the root cause of the problem. So we want to, and the cancer cell is addicted to this, because in the lab, if we develop experimental techniques to shut that switch off, the cancer cells die. It's called tumor addiction. The tumor's depending on this mutation. Now, when we've sequenced a lot of cancers, there may be mutations in the cancer. Some are passengers and some are drivers. We want the drivers. So there's been a lot of medications now targeted against these mutated receptors and other mutated targets that are part of targeted therapy, and it's changed the future of oncology. And I want to give you some examples of that because it's just beginning. It's specific for the cancer cell. It hits the target to which the cancer cell is addicted, and it does not engage normal cells. Because remember, the normal cells have a normal genome. It's the cancer that's corrupted. So I want to tell you what a biomarker is, because we use this term all the time, but what is a biomarker? A biomarker is a gene or protein measured in the tumor or blood that may indicate the presence of cancer, so you can use it for screening. It may determine the subtype of the cancer, so you can classify it better. There's no doubt now that breast cancer is probably about 30 to 50 different diseases. We may call it breast cancer. We may divide it 3 ways now, but it can be divided 50 ways if you look at it with using genetics. This biomarker may indicate the aggressiveness of the cancer. Could it be prognostically? So how difficult is my tumor to treat? And finally, what I've just got through talking about, it can be a target. And since I treat cancer, I'm really interested in those that relate to therapy. But a biomarker can be anything. So when we talk about personalized medicine, we may use it to screen for things. I want to give you one example of that. It may determine the subtype of the cancer to allow us to pick better therapy. Again, HER2/neu amplification is one great example. It can determine the aggressiveness of the cancer, so we can plot out therapy in terms of how aggressive we need to treat it, and it's a target. Any questions about what I'm telling you so far? Yes?
PSA is a biomarker?
So PSA is a biomarker. So the prostate-specific antigen is a biomarker. It's not a great biomarker. And then we can talk about some of these because the problem with biomarkers now is that it may pick up disease that may be harmless and lead to overdiagnosis. It may not be sensitive enough, so you may not pick up aggressive lesions. And it may be, the value may be such that you have to screen, 3,000 patients to benefit one. And that's the whole debate in the papers, and we can talk about that. I'm going to talk about certain genetic biomarkers that I think have significant advantage. Okay, so the vision now is that using this genomic information I've just provided the outline for, can we do an integrated biological analysis of the tumor as well as the patient to identify known targets in their own cancer and discover biological subtypes that we may have not realized before? Most importantly, can we select optimal therapy for that patient? So for this one patient, what's going to benefit their tumor the best without causing unnecessary side effects? And obviously, we want to improve outcome, minimize side effects, and reduce costs in the final analysis. With a $3.2 trillion budget, we want to cure cancer and we want to eradicate diseases like diabetes, but we will suck all the money out of the world unless we do this better. So the strategy for us at the NYU Cancer Institute is to develop the infrastructure and technology to identify these relevant lesions in individual patients, to develop standard procedures to how we biopsy tumors and analyze them. The instrumentation now has been breathtaking. So it's difficult for me to believe, after being a postdoc at Stanford, that I've just sequenced the full genome of 20 children with acute leukemia. And I've done that within a year. It took the NIH 6 years to sequence one genome. We can do this now probably in 3 days, and the cost is going to come under $1,000 within 3.5 years, we estimate, because the instrumentation now has changed everything. We want to identify targets, but discover new ones as well, these biomarkers of the future. And most importantly, we want to develop educational tools to educate the public, as well as physicians, about how to use this information to guide a therapy for the individual patient. Because I can tell you now, the field's moving so rapidly that even the most up-to-date physician can't keep up with all of this. So when we develop targeted therapy, just to go over this, we want to develop a target that is in the tumor and not shared by normal cells. The target has to be essential to the maintenance of the cancer. And I want to just tell you about cancer stem cells, because this is a topic that you may have heard about. When a patient with cancer is first diagnosed, they have about 10 to the 12th cancer cells in their body, so about a kilogram of cancer cells. And that population, which is 10 to the 12th, large number of cells, not every cancer cell is identical to one another. There may be a small subset of those cancer cells, about 0.1%, that's like the seed that leads to the bulk population. It's like the mothership. It's called a cancer stem cell. So there are diseases now where we can eradicate the bulk tumor, but we haven't eradicated the stem cell. And that stem cell provides a reservoir for that tumor to come back. So when we talk about targets, we want to make sure it's in the bulk population, but also in the stem cells that may act as seeds. They could lead to recurrence in the future. Now we want to make sure that we pick on a pathway that doesn't have a twin pathway that the cancer cell can shift tracks and go to. It's called redundancy. So Mother Nature has developed a number of redundant pathways. So if you damage one, another one can take its place. And under normal circumstances, this is an advantage. But for cancer cells, we don't want them to change tracks and evade what we're doing. So we want to pick a pathway that's one of sort of the nodal points that is really one of the essential ones. So it can't switch tracks. And importantly, we want to develop agents that get access to the target. It doesn't help us to develop a new agent against a brain tumor if that agent can't penetrate the blood-brain barrier. Okay, so these things have to be armed in such a way or put on cargo so it can get to the tumor. So the field really changed with this lesion right here called the Philadelphia chromosome. How many people know about the Philadelphia chromosome? Or a drug called imatinib, or Gleevec. So chronic myelogenous leukemia is a disease of white blood cells, and it leads to proliferation of white blood cells in the bloodstream. Leukemia means white blood, and patients with CML go through a chronic phase where they can live with their disease, but then it degenerates into an accelerated phase in acute leukemia. It wasn't cured by anything until 2001, with the exception of bone marrow transplant. Now, in 1960, when chromosome structure was first being identified, Nolan Hungerford in Philadelphia looked at CML cells from patients with CML and noticed that they had a funny-looking chromosome called the Philadelphia chromosome. It's right here. And it was abnormal. It was in the CML, but wasn't in the normal cells. Now, that turned out to be really a swap in genetic information between chromosome 9 and chromosome 22. This is that translocation I was telling you about. And it led to this thing called BCR-ABL. This was a chimeric protein generated from this abnormal swap between chromosomes in CML. It's seen in 100 cases, it's seen in 100% of CMLs, this Philadelphia chromosome. So investigators determined that actually what is happening here is this ABL gene on chromosome 9 is one of those growth-promoting genes I told you about. It's called ABL. It's called a tyrosine kinase. It's responsible for taking phosphate groups and activating targets. So it's on and the cell is growing. And what happened is that this switch, this chimeric protein now, took on properties so that the ABL switch was always on. And this thing was taking the phosphate group from one protein, putting it on a substrate, and activating a growth chain. So what people at Novartis did was, this is the structure of this protein. This ribbon diagram here is a fancy way of drawing this. And it has a pocket here. And this pocket is what it used to take the phosphate from one protein and transferred it to another. It's a critical part of this protein. So they screened a library of compounds to see what compound could stick itself in this pocket and prevent BCR-ABL from working. Now remember, BCR-ABL is only in the cancer, and the compound they came up with would hit BCR-ABL, but not the normal ABL in normal cells. It was tumor specific. Now patients with CML who didn't have a donor and who failed all therapy were dying of their disease. And I remember at a conference in 2001 in San Francisco at the ASH meetings where Brian Drucker from Oregon announced the first results of the clinical trial for patients with refractory CML using this agent. And the benefit of this agent is it's an oral medicine. You don't get an IV. You don't lose your hair. It doesn't lead to low white blood cell counts. In fact, you know what? The way drugs are developed in cancer therapy is you go through what's called a phase 1 trial. And the purpose there is to give the drug and determine the maximum tolerated dose, the dose that you can give without causing untoward side effects. When they did that for this, it used to be called Gleevec, now it's imatinib, they never got a maximum tolerated dose. It was dramatically different from the kind of therapy we delivered. It hit the cancer, didn't have the side effects, you took an oral pill. Now, this is the data he showed, and I can tell you here, there was at least about 25,000 people in the audience, and you could hear a pin drop. Because of patients with refractory CML, 95% of them went into a complete remission. That means that their leukemia disappeared from the blood. Okay? They went into remission. Now, some of those still had the Philadelphia chromosome when you look by microscopic techniques, so the clone wasn't eradicated completely. But these were patients with refractory disease. Now, we're giving it to patients with disease initial diagnosis now, and patients are living with this forever. So the patients are staying on this drug now, not going to transplant, and it's changed the disease entirely. However, a small percentage of patients will relapse on imatinib. And the question is, why do they relapse? And when we and others have analyzed the BCR-ABL, the target in the relapsed cancer, what we found is the cancer develops mutations in this pocket now, so it no longer binds imatinib. So it has gone around the imatinib. Now the good news is now there are second and third generation BCR-ABL inhibitors, dasatinib and others, that will overcome all these mutations. So the pharmaceutical companies and academia has kept up. So dasatinib now, and is a third generation now, that not only is 300 times more powerful than imatinib, but it overcomes all the imatinib mutations with one exception, and it happens to hit another pathway that is essential. Now, I will tell you that for patients with CML, they, for the most part, can't stop taking the medication. So we probably have not eradicated the stem cell, but the disease does not come back. Some patients have gone off it for the most part, but when they go off the drug, it comes back. So in this case, we want to develop another imatinib or dasatinib that's more stem cell specific. But thankfully, we've changed this disease significantly. Now, this is the best known target, BCR-ABL and CML, but there are a lot of other targets now that have surfaced that are just like BCR-ABL and CML. I'm going to tell you about BRAF in melanoma, the EGFR mutation in lung cancer, this ALK fusion in non-small lung cancer, KRAS in colon cancer, and of course, HER2 new application breast cancer. These tumors need to be screened for these mutations because if you have them in your cancer, you will respond to these drugs that have just been approved. This is the frequency of that target in the population. It varies from 95% for CML to as low as 5% for the ALK fusion in lung cancer. Now, I just want to point out one thing. One of the things that we have to change in cancer treatment is the length of time it takes to develop a new drug. So this BCR-ABL target was really discovered in 1960, and it took till 2001 to develop a drug. I can't tell you that the ALK fusion for lung cancer was discovered in 2007, and the drug for that was developed and approved by the FDA within 3 years. A remarkable advance. So those of us in academia, in pharmaceutical corporations, and the FDA recognize that we need to change some of the regulatory requirements to compress the length of time for drug development, because our patients can't wait 12 years to develop a new drug. That's the average time it takes to develop a new drug. But I want to talk about one more thing, and it's changed the way we've been able to advance cure rates for cancer is by knowing what works best for everybody. But then as new developments take place, we want to determine if the new drug has any advantage over the standard therapy. So everybody, it's called a clinical trial. This is cancer chemotherapy now, so you can't just indiscriminately give it to everybody. But half the patients get the best available therapy, and half the patients get the best available therapy plus the new drug. And so everybody gets the best available therapy. And this new drug, it may cause side effects or it may help. The problem is that in order to develop these randomized trials, we look for outcome for the whole population. If I study 100 patients and the percent of patients that have my target is only 8% of the total, I won't pick up that signal in a large randomized trial, right? It's going to be for those 8% of patients, that drug is curative. But since the other 92% of patients don't have that target, I may not have a trial big enough to pick up that signal. So the whole point now is that we got to pair the trial with the genetic lesion that the patient's cancer expresses. So it's changed everything. So what we're going to do now, probably, instead of these large randomized trials, do much smaller trials where we identify the target and this patient's eligible for a new agent that hits that target. Now, I want to just talk about BRAF. I went to a conference about 6 months ago at the NCI. And as you know now, patient advocates are really a public voice in NIH and NCI research. And this was a lung cancer conference, and a woman got up who talked about her experience with lung cancer. And she was soon to be a grandmother. She was about 55, 56 years of age. And she went to her doctor because of a chronic cough. A chest X revealed a lung lesion, and a biopsy showed non-small cell lung cancer. The first doctor she went to told her she should go home and get her affairs in order because she had 6 months to live. The second doctor she went to told her pretty much the same story. She didn't like that answer, and she wanted to live long enough to see her grandchildren. So we went to, actually, I think it was a fourth doctor who said, you know, there's this new target called the ALK fusion that was just discovered. It's only in 7% of lung cancer patients. It tends to be seen in non-smokers, by the way, so there is some hint that based on history you could pick this out. Let's screen your cancer for that fusion. Remember, this was the fourth doctor she went to. They screened her for that, and she had the ALK fusion. She went on crizotinib for a clinical trial, and she is living 5 years later on this drug. So it just goes to show you the profound impact. This happens to be melanoma here. This is actually melanoma. I was talking about lung cancer. This is BRAF and melanoma. So 60% of melanomas have a mutation in this gene called BRAF. It's a growth gene. And you can see here this patient with advanced melanoma with no other options. This is a lung, this is a CT scan right here. This is the backbone. This is a slice this way, right? So this is the heart. This is a fluid around the heart. These are the lungs right here. And you can see all these dots here represent metastasis from the melanoma. This individual had a liver metastasis. Well, within 8 weeks on this drug here, this Plexicon drug, these metastases melted away within 8 weeks. If you do a stain of the melanoma before and after, these stains are, all this brown here, are surrogate stains for the melanoma. You can see that within 15 days, this stain is for the growth signal I told you about. So the BRAF activates something called ERK. You can see that growth switch is switched off within 15 days of taking the drug, and the tumor is gone within 8 weeks. Okay, this is the Plexicon drug for BRAF. This has just been approved by the FDA, I think about 9 weeks ago. Another example. Now, these patients also recur. But when they recur, they don't occur with mutations in the BRAF. They occur down here. So this is called an off-target mutation. They occur in this MEK gene down here. So current trials for melanoma are aligned so that you use the BRAF drug, but also throw in a MEK drug. So it's combination molecular therapy. It's preventing the cancer from developing the resistance. Just one example to how we can't probably depend on a single molecular drug to cure cancer. It may be many. And if you think about it, there are 3 types of cancer. There's sort of dumb cancers like CML, where one drug hits the target and that thing goes into remission. There are intermediate tumors like the small percentage of lung cancer patients and these melanomas where you hit it with a drug, it goes into remission for about 6 to 8 months, and then it comes back. We need to do a little bit better with those. And there are some tumors like pancreatic cancer, and I'll finish with that, that are still not giving us great examples of targets that we can attack just yet. But I just want to get back of, this is a list of targets that are available. Now what we're doing now at NYU, and I'll finish with the next slides, is developing a center for advanced diagnostics and personalized medicine. And our vision is that every tumor that we treat will be fingerprinted for these mutations that will allow us to develop better therapy for that individual patient's tumor. And what we're going to do here is that, I put it like a lab slip. We want to get results that will tell us whether that patient's eligible for approved drugs that will hit their target. BRAF and the ALK fusions are examples. There are lots of new agents coming down the pike in clinical trials. We want to detect those to put those patients on clinical trials where these new drugs may not be yet approved, but they're promising enough to make sure we get those patients on clinical protocols. And finally, we're developing a large warehouse where we aim to sequence the whole genome of cancer cells to develop a comprehensive genetic portrait of lung cancer, melanoma, at the fine detail of the whole 3.2 billion base pairs. So we're developing, in addition to what we're doing in-house here, like I told you, I'm a San Franciscan, and I was told about the competitiveness of New Yorkers. And we are a very competitive bunch. But I can say that in our city, in contrast to other communities, that all the major institutions in this city are partnering around a proton beam center, but also around a genome center. So all of us have partnered together, ourselves, Sloan Kettering, Mount Sinai, Cornell, Cold Spring Harbor, and we're going to use the new instrumentation in high throughput sequencing. So remember, we're going to do a snapshot on the tumor in-house to detect 20 of those relevant mutations. But we want to go through the whole, we're going to come together in something that's going to be, the ground's going to be broken on, I think it's November 3rd, into, with the express purpose of accelerating new discoveries, diagnostic treatments for human diseases, mostly cancer, by leveraging the resources of the leading academic centers in this city to develop this center. And I'm very excited by this, because we aim to lead the genomics revolution at a scale and a speed that helps our patients. Now, I want to end with one final vignette. I told you about pancreatic cancer. Now, there's no doubt we are focused on treating advanced cancers. Now, I told you about, we've been able to sequence the full genomes of, in my lab, childhood leukemia. They've done pancreatic cancer. And cancers themselves don't have a higher mutation rate, right? They're not hypermutable. They're just mutating, and you happen to hit the wrong target, and that endows that cell with a growth advantage. So by using the known mutation rate of human cells and determining the number of mutations in a cancer cell, you can begin to estimate the lifetime of that cancer. Now, for pancreatic cancer, that is at this moment in time, the best way to cure pancreatic cancer is to catch it early and to surgically remove it. But we have no great biomarkers for pancreatic cancer. We can't detect it at its silent stage. It doesn't become manifest until the patients develop symptoms, and by that time, the disease is far advanced. When they've calculated the lifespan of a pancreatic tumor, using this evolutionary biologist from Harvard, it's estimated that the average pancreatic tumor has been there for 12 years. And that the mutations that has led to its ability to metastasize and grow has only happened over the last 2 years. So the goal now is to use this genetic information to pick up cancers not at the advanced stage, but the early stage. Now, I told you that pancreatic cancer has a mutated RAS gene 90% of the cases. And we have technology now to pick up a mutated RAS in a stool sample. It's called digital PCR. And so the idea here is that we could probably screen for pancreatic cancer now, looking for these molecular markers in serum, which is not quite as good as stool, to pick up pancreatic cancer at its earliest stages. Now, the problem is, as I told you, having a RAS mutation by itself doesn't mean you have cancer. Because in many of us, remember, these mutations are occurring all the time, and you don't get the next 4 steps. So we don't want to overcall things and lead to unnecessary screening and things like that. But I am optimistic now that using some of this molecular technology, that we can develop more precise biomarkers, because we're looking for a mutation now. With PSA is sort of just a protein, whereas this is really a biomarker. So I think in looking forward, using this technology to develop better prevention strategies may in fact be another mother lode of advances for childhood cancer therapy. Now look, that's a lot of information. I want to stop now and answer any questions. Thank you.